independently transcribed in yeast, of course. So what we know for now is the basic model of snoRNA, the CD-box snoRNA transcription, and it can be terminated at two different termination sites. The first site here, transcript is terminated by NRD1 NAP3 complex. Then the transcript is oligoadenylated and then polyadenylated, and after that it's trimmed by exosome. And if the transcript is terminated at the second termination site, it has a special mRNA-like signal for termination, and then it's polyadenylated by PAP1 enzyme. And if it comes to 5' end, its maturation is like it's cleaved by RNT1 enzyme and then exonucleolytically trimmed by RAT1 enzyme. What we recently found and what I wanted to prove in my study is that these two machineries responsible for 5' and 3' end maturation are coupled. So the processing of 5' and 3' end is dependent on each other. So we based our model on previously obtained results in different labs, and they are as follows. Factors involved in transcription termination and formation of mRNA 3' end associate with the promoter region. It was published by Topey-Zirowicz in 2011. Then what we know is that cap binding and NRD1 NAP3 SEN1 complexes co-purify, so that suggests interaction of machineries acting on both snoRNA ends. That was published by Vasilieva in 2006. And then some research from our lab is that interaction between CBC and NRD1 NAP3 complexes is direct. Then CBC remains associated and snoRNA genes until transcription termination. And the last thing is that RNT1 enzyme, this endonuclease, is recruited to maturating snoRNAs at late stages of transcription. So the aim of my study, basing on the previously presented statements, is that processing of 5' and 3' snoRNA ends in Saccharomyces cerevisiae is coupled. And I wanted to check this hypothesis doing the following analysis of snoRNA 3' and 5' end stages in mutants with defective end formation, then description of snoRNA synthesis defects in mutant strains, and the last thing was the characterization of alternative 5' snoRNA formation mechanism by DCP1, DCP2 complex in the absence of RNT1 cleavage. And the materials I've used, the first thing is yeast strains that were used in my study. All of them are mutants, deletants of 5' and proteins that process the 5' end. RNT1, that is homologous to bacterial RNase 3, double-strand specific endoribonuclease, and it functions in the 5' end of CD-box snoRNA. The next enzyme was TGS, it's nuclear trimethylguanosine synthase, responsible for MMG cap hypermethylation to TMG cap of snoRNA. The next thing is CBP-AT protein. It's a nuclear cap-binding protein, and together with CBP20 are subunits of the cap-binding complex. And the last thing was DCP1, DCP2 complex, responsible for rapid RNA decapping by removing the 5' cap and leaving the 5' end susceptible to exonucleolytic degradation. And in my study, I focus on a few snoRNA molecules. All of them were independently transcribed box CD snoRNA cleaved by RNT1 in this specific motif, SNR 68, 64, and 65. And additionally, I also take a look at SNR 30 as a control, because it's also independently transcribed CD-box snoRNA, but it has TMG cap, so it's not processed at 5' end. Now I can proceed to my results. At first, what we confirmed is that 5' snoRNA maturation defects lead to the accumulation of 3' end precursors. So what we did was, was the Northern blot analysis for SNR 68 and 64 in wild type and mutant strains, and I did the hybridization after NNase treatment. in the presence of oligocomplementary to the mature snoRNA. It was placed around 100-200 base pairs upstream of the 3' end. So here it was cleaved by RNase H. So what we could do was the observation of 3' end extended precursors. And here what we can see is that in mutants of RNT1, the 3' precursors accumulates, and it's visible that for double mutants with TGS the effect can be strengthened, but for this double mutant with CBP-AT it looks like the accumulation is lower. It was very similar in SNR68 and 64. The next thing was that I did a CRT-PCR analysis, and the total RNA was treated with RNase H to cut the cap, and thanks to that we can ligate the molecules, then reverse transcribe them, and take a look at the accumulation. So what we found is that in mutants with RNT1, the accumulation of 3' precursors is visible, and again in the double mutants with TGS the accumulation is even higher, and in this mutant it's lower. And it's the map of SNR68 mature molecule and the whole precursor, and here are some oligos I've used. After CRT-PCR, we also took some products and do this sequencing. So the interesting thing was that we couldn't obtain the full-length precursors of these mutants of RNT1. However, for this wild type for RNT1 strains, the products we obtained were very long. We assumed that it may be the full-length precursors that didn't overcome the processing yet. And the last part of my presentation is the proof that there could be an alternative maturation pathway, and that this DCP1, DCP2 complex plays a role in this pre-snoRNA processing. The first experiment was that I checked the accumulation of 3' extended precursors in the capping mutants, so diletants of DCP1 or DCP2. And what we can see is that for SNR68, the precursors accumulate in all diletant strains. However, for SNR64 and 65, the accumulation was not visible. So that the effect is visible only for this molecule could be because differences in dependence of RNT1 cleavage. That's our theory, but it needs to be proved. The next thing I did was checking the accumulation of SNR68 precursors in RNT1 and DCP2 mutants, so in double mutants. We tried to obtain a strain with these two proteins deleted. However, it wasn't possible because this strain is lethal. So what we tried to do, and we did, was constructing a strain. Was constructing a strain with plasmid carrying SNR68 molecule with wild type or mutated recognition motif for RNT1, so not cleaved for mutants, not cleaved by RNT1. And the mutation looks like this. It was this change, and what I saw was that for these strains where the plasmid carried the molecule with mutated motif, the accumulation is visible, and additionally in DCP2 deletant with this plasmid carrying a mutated motif, the accumulation is also visible. And after that, I also checked the 5' end extensions in RNT1 and DCP2 deletants. And sequencing after CRT-PCR gave me results that 90% of precursors accumulated in DCP2 deletants and are not cleaved by RNT1 have shorter 5' ends than observed in RNT1 strains. That is very surprising and interesting result. So the possible explanations are that alternative transcription start site is present, and alternative decapping enzyme is present, like DXO or something else, but we are not sure for now. And to conclude, all results I've presented lately is that defects of 5' snoRNA and processing, especially lack of RNT1 cleavage, lead to inefficient 3' end formation accumulation of extended precursors. The next thing is that this phenotype is additionally modulated by mutations of other proteins acting at snoRNA 5' ends. It's partly rescued by this absence of CBC, like CBP18 protein, and additionally strengthened by deletion of DCP1, DCP2 complex or TGS1 protein. And the last conclusion is that synthesis of mature snoRNAs in the absence of RNT1 cleavage suggests the existence of alternative maturation pathway mediated by this DCP1, DCP2 complex and independent of RNT1 enzyme. My acknowledgement to RNA Kufel's lab, and thank you very much for your attention.
Questions, please.
Okay. So thank you very much once again.
You have a beer on the last slide, so you're not drinking a lot of beer after your work.
Yeah.
Okay, thank you.
Thank you.